and welcome everybody. Good evening, good morning. Welcome to the best of ATS 2025 Pulmonary Circulation Assembly, the translational abstract hosted by the ATS PC Assembly. And today we have a lineup of 5 presenters present our excellent abstract from the last ATS. And the discussion will be fueled by a diverse and a truly transatlantic panel of discussants, including Elena Koncharova, who serves as a Director of Pulmonary Vascular Disease at UC Davis, and who has a focus on molecular and cellular mechanisms of pulmonary hypertension, by Allan Lawrie, who is a Professor of Pulmonary Vascular Disease at Imperial College in London, United Kingdom, and by Roham Zamanian, who is a Professor at Stanford University, who specializes in the treatment of PH and is a leading expert in clinical trials. My name is Jurjan Amon from the Amsterdam University Medical Center Amsterdam and the Netherlands, and I'm co-hosting this session with Mariam Sharifi from Pitt University, who will announce the first speaker.
Hi everyone. Yes, I'm Mariam, and along with Dr. Aman, we will do our best to help facilitate this seminar. So as he mentioned, we will have 5 presenters, so please hold your question until the end of each presentation, at which point you can either raise your hand or put your question through the message. Thank you for our panelists again to provide the time. And with that, our first presenter is Christine Farrell from University of Colorado.
Thank you to the organizers and the moderators. My name is Christine Farrell. I'm one of the Pulmonary and Critical Care Fellows at the University of Colorado, and I was hoping to chat a little bit about a project that I've been doing during my fellowship. And so at the CVP at the University of Colorado, we've been very interested in inflammation as a driver of pulmonary vascular remodeling and pH. Previous work from the CVP has shown that the degree of inflammation in the perivascular space correlates both with vascular remodeling and tends to correlate with mean pulmonary arterial pressure. What's a little bit less cited from this paper is that 50% of all of the vasculature in group 1 PAH patients also had microthrombi within them, and we've been particularly interested in the intersection between thrombosis and inflammation. Particularly, we've been interested in complement as a potential link between these two processes. Patients with PAH have increased complement activation and deposition within their lungs in the perivascular space compared to healthy controls. And then in preclinical models, mice that lack C3, a main component of the complement pathway, have attenuated hypoxia-induced RVSP, attenuated hypoxia-induced RV hypertrophy compared to wild-type controls, and then also have decreased platelet activation. In hypoxia shown in black here, there's generally increased platelet activation as shown by platelet expression of a molecule P-selectin. And in mice that lack C3, they have prevented platelet activation from hypoxia. And so looking a little bit at the complement cascade, there's 3 main inputs that converge on this molecule C3 that is then cleaved into C3a that I've been particularly interested in. It's an anaphylatoxin that binds to its receptor C3AR, which is present on various immune cells and on platelets. And then it also goes further down the complement cascade to cleave C5 into the anaphylatoxin C5a, which binds to its receptor C5AR1, which is also present on various inflammatory cells and on platelets. And work from the CVP has previously shown that there's increased expression of cells that express the complement anaphylatoxin receptors C3AR1 and C5AR1 in the perivascular space of mice and rats that are exposed to hypoxia. However, what cell types express these receptors is currently not known. And so my 3 main research questions is there increased C3a and C5a generation in response to hypoxia? And if so, is the interaction between these anaphylatoxins and their receptors important for the development of hypoxia-induced pulmonary hypertension? And if so, what cell types express these receptors and mediate this protection? And so to study this, I've been using the murine hypoxia model, where I've taken wild-type mice, whole body C3AR knockout mice and whole body C5AR1 knockout mice, and either kept them at sea level or exposed them to hypoxic conditions at 18,000 feet, which correlates to around 10% FiO2. I then look at around 3 days, looking at markers of inflammation in the blood and in the lungs of these mice, and then also carry them out to 21 days to look at pH endpoints. And so for my first research question, is there increased C3A and C5A in the plasma of these mice? The answer is yes, however, at different time points. If you take mice and then expose them to progressive hypoxia, C3A tends to peak in their plasma at around 3 days. However, C5A tends to peak a little bit earlier, around 5 hours, and then goes back to normal levels or even somewhat less levels than normoxic mice. And we think that this is because C5A is either being taken up by various cells or actively degraded. Also looking in the plasma, in general, platelets have low level expression of the C3AR and C5AR1. However, if you look at them around 3 days, they tend to upregulate their expression of these receptors on their cell surface. And you may be wondering, how can you generate C5A in the absence of C3A based on that schema I just showed? It's been previously shown, and we confirmed this, that you can get C5A generation even without C3. We took whole body C3 knockout mice and exposed them to hypoxia and showed that there was an increase of C5A generation in these mice that lack C3. And one of the proteins we think that may be responsible for this is thrombin, which has been shown to be able to activate C5. And these C3 knockout mice do have increased levels of thrombin in their systemic circulation compared to wild type controls. And so for my second question, is this relevant to pulmonary hypertension? It appears that it is for the interaction between C3A and C3AR. I took whole body C3AR knockout mice and exposed them to 21 days of hypoxia, and they have prevented increase of hypoxia-induced RVSP compared to wild type controls. And then they also have attenuated increase in right ventricular hypertrophy as measured by Fulton's index in these mice compared to wild type controls. In addition, they have partially protected from pulmonary vascular-induced remodeling. They have prevention of the increase in medial wall thickness compared to wild-type mice. However, if you look at vessels less than 50 microns, there's no difference in small vessel muscularization in these mice compared to wild-type controls. In contrast, looking at whole body C5AR knockout mice, these mice are not protected from hypoxia-induced pH. Their increase, as shown in blue here, from normoxia to 21 days of hypoxia is very similar to their wild-type counterparts in both RVSP, in terms of right ventricular hypertrophy as measured by Fulton's index compared to wild-type controls, in terms of their medial wall thickness, and in terms of their small vessel muscularization. And so this led me to what cells might be expressing the C3AR receptor and mediating this protection. When looking at single cell sequencing data of mice and humans, the two most common cell types in the lungs that express this receptor are macrophages, with IMs being the most abundant, AMs down below, and then platelets as the second cell type. And so we looked at both of these. We looked at their total numbers at day 3, and it looks like they are not protected from hypoxia-induced increase of IMs at day 3 of hypoxia exposure in these mice, as also seen in wild-type controls shown here in left. And then platelet numbers have a trend towards being upregulated actually at baseline in these C3AR mice, and then do not further increase with hypoxia exposure. So we do not think it's related to the absolute number of trafficking of IMs or platelets into the lungs of these mice. We also looked as platelet activation attenuated in these C3AR knockout mice. We did not see an increase in response to hypoxia of platelet expression of P-selectin, which is shown to be a marker of activation as compared to wild-type mice. However, whether or not this is clinically relevant is not 100% known. P-selectin binds to PSGL1 on the surface of leukocytes to mediate platelet leukocyte aggregate formation. And when we looked at platelet leukocyte aggregate formation in these mice, there was no difference in normoxia and hypoxia compared to wild-type controls, looking at all leukocytes and then also particularly at monocytes. And so in conclusion, C3AR but not C5AR1 mediates hypoxia-induced pulmonary hypertension. However, this is not done by alternating cell numbers of the two cell types that most commonly express this receptor. And so for future directions, I'll be looking if the phenotype of these macrophages and platelets in these C3AR knockout mice is different, and maybe these are less pro-inflammatory compared to wild type controls, and it's more of a phenotype, not a number change. And then I'll also be looking further downstream on the pathway at whole body C5 knockout mice to see if this may also be mediated through the MAC complex. And so I just wanted to thank my mentors and the members of my lab for all their assistance in this project, my research advisory committee, and my fellowship program for the support. And I'll be open to any questions.
Thank you. This is wonderful presentation, and this is very intriguing data. So congratulations with this. I am very interested in your data on C3AR chaos mice. So, pretty much, if I understand correctly, what you show that this you have RV hypertrophy existing without changes in SRVP. So would it imply that there is direct effect on RV morphology and probably maybe function that is independent of vasoconstriction? Can you please comment on this?
Yes, Elena, we had the same thought. I've actually been looking at the RVs, looking at kind of their cardiomyocytes. So that's kind of more to come to see. There's not been a whole lot of data at all about the interaction of the complement system in the RV macrophages in particular. And so I think this is something that definitely needs to be explored. And I'm just starting to dive into that.
So, sorry, Christina, if I can just jump in and ask a follow-up. So I think it's really interesting. Thanks very much for presenting it. And I'm glad to see there was a lot more in there than was the abstract. I think you've done a huge amount of work there. I guess one of the things that we're kind of interested in, and I'm glad Elena mentioned the RV, because obviously the RV in particular over the LV has been shown to express a lot of the C3 molecule itself. But I wonder how much of what you're seeing do you think is specific to a hypoxic effect, or how much of it is more of a pathogenic effect? And we're limited with the mouse in terms of what we can throw at it to induce pH, but I just wonder if you could maybe firstly answer that, and then secondly, maybe just comment on where you think the C3 is coming from. Obviously, it's predominantly produced by liver, but there's lots of evidence, including from the kind of Stenmark and Tudor paper, suggesting that C3 is also expressed locally. So I just wonder if you could comment on that.
Yeah, the second one is, I think, easier to answer. There's some thought that there's this cellular store of C3a, particularly from fibroblasts. And so that's kind of been a question for us, is what cell type is releasing this and how do we best get at that? I think Kurt Stenmark is definitely most suspicious that it's actually getting released by the fibroblasts in the lungs and acting locally. That being said, we're also seeing it in the systemic circulation, so probably some components coming from the liver as well. Platelets themselves do store complement inside of them, and when activated can release some of this. And so we've also been kind of wondering, are platelets kind of a reservoir for some of this in the systemic circulation as well? And whether what's kind of going on with the first question, that is a hard one for me to be able to answer. We know that mice of C3 and C3AR knockout mice do have a kind of bleeding phenotype. And so kind of where this is mediating protection in response to hypoxia is not entirely clear to me at this juncture.
Okay, great. Thank you.
Kristen, I had a question, quick one. Do you see any cognitive issues with these mice?
There has been some data in the stroke literature and then also in about kind of C3AR and trying to prevent it for healing of a damaged brain. I have not noticed personally in these mice any cognitive issues. I know that other groups have looked into this for different models such as schizophrenia and things such as that.
So our next presenter is Amber Mercevy, if I'm spelling that correctly. And she's from the University of Pennsylvania. Amber, please take it on.
Thank you for having me today. My name is Amber Messervey. I am a fellow at the Hospital of the University of Pennsylvania, and I'm presenting some of my fellowship project work as well today. This work is titled Plasma Proteins Differ by 6-Minute Walk Distance in Physical Activity in patients with pulmonary arterial hypertension. So there are a number of potential applications for biomarkers in pulmonary arterial hypertension, or PAH. In this study, we were interested in identifying biomarkers which correspond to measures of exercise limitation. Exercise limitation is an important measure of disease status and is predictive of outcomes in pulmonary arterial hypertension, and it's measured using clinic-based assessments. There's currently no serologic correlate. So we hypothesized that a biomarker corresponding to exercise limitation could be used to augment risk stratification, could be used to aid in disease monitoring, potentially remotely, and also could be used to assess treatment response. So with those goals in mind, our specific aims were to determine whether levels of specific plasma proteins were associated with baseline 6-minute walk distance in a clinical trial population of patients with PAH. And then further, we were interested in seeing whether we could extend this into the physical activity measures we had from actigraphy data. We conducted this study using data collected within the context of the pulmonary hypertension and anastrozole or PHANTOM trial. This was a phase 2 randomized clinical trial of 84 adult patients with PAH across 7 U.S. centers. Anastrozole did not improve the primary endpoint, which was 6-minute walk distance at 6 months. However, the study did collect plasma, 6-minute walk distance, and physical activity data at 4 time points across 1 year. I will say the data I'm showing today is really just the baseline data, however. For context, plasma samples were run using the Olink Cardiometabolic Explore panel, and 7-day activity was collected using a Warren actigraph. These devices collect a number of measures, one of which is vector magnitude, which is the raw triaxial movement that is then used to categorize activity and also to derive step counts. So we analyzed the data using multivariable linear regression, 6-minute walk distance, and vector magnitude as a function of protein levels. We adjusted for age, sex, PH etiology, and wear time, and we did use an FDR-adjusted p-value of less than 0.05 across all analyses. Shown here are the baseline patient characteristics. So the average age was around 60 years old, about 60% of the cohort female. You'll see that almost half of the patients had either idiopathic or heritable PAH. About 80-90% had WHO functional class 2 or 3 disease with a mean starting 6-minute walk distance of around 430 meters. And almost everyone was on either dual or triple PAH therapy. So shown here is our baseline protein expression by 6-minute walk distance analysis. To orient you, on the y-axis is significance, so those proteins higher up on this plot are more significant, and on the x-axis is effect size between protein level and 6-minute walk distance. So those proteins shown in orange here were significantly higher for lower baseline 6-minute walk distances. I just want to point out a few of the proteins that you may be familiar with. So NT-proBNP and NPPB is BNP. So those were significant, though their effect size was small. And the protein that I'm focusing on here today is collagen 18 alpha 1, also known as endostatin, as that had the most significant relationship with baseline 6-minute walk distance. We did extrapolate out that for every 1 standard deviation increase in endostatin at baseline, there was a roughly 47-meter decrease in baseline 6-minute walk distance. I don't know if my p-values are hidden here, but these are all significant. We did see external validation of this association between endostatin and exercise capacity using data collected from 47 patients with incident connective tissue disease, PAH, who were enrolled at the Sheffield Pulmonary Vascular Disease Unit in the UK. Importantly, these patients had their proteins measured on the Myriad Discovery Map platform, and their walk test was completed using the incremental shuttle walk test. So we did apply z-score transformation to all variables in both cohorts before analyzing these. And we found that in the Sheffield cohort, for every 1 standard deviation increase in log protein concentration of endostatin, there was a 0.38 standard deviation decrease in log incremental shuttle walk test, which was significant. Very similar to what we found in our own cohort, every 1 standard deviation increase was associated with a 0.4 standard deviation decrease in 6-minute walk distance. So we use this to say we did externally validate this association despite the difference in patient cohort, protein modality, and walk test modality. So what is endostatin? It is a circulating peptide and cleavage product of collagen that's in the basement membranes of blood vessel walls. It's known to be a potent inhibitor of angiogenesis, which acts on pulmonary artery endothelial cells and vascular smooth muscle cells. And higher circulating endostatin levels have been associated with a number of adverse outcomes in PAH, including worse hemodynamics, reduced RV function, higher risk adverse outcomes, and enhanced prediction as well. So we're excited that our findings aligned with what's in the literature, and we wanted to see if we could extend this into our physical actigraphy data that we have. So this is our baseline protein expression by vector magnitude analysis, same volcano plot. We found a lot of the same proteins as we saw in the 6-minute walk distance, which perhaps is not surprising. And again, endostatin showed the most significant association here. We found that for every 1 standard deviation increase in endostatin at baseline, patients had roughly 30 minutes more time spent sedentary per day. Again, this is looking at step count, so a lot more noise here, but we see this relationship between endostatin and step counts as well, with every 1 standard deviation increase in endostatin being associated with almost 1,000 fewer steps taken per day. Lastly, we used a Cox proportional hazard model to assess clinical worsening over time as stratified by baseline endostatin. So those patients that had at least 1 standard deviation above the mean baseline endostatin, shown here in orange, had significantly more clinical worsening events over the course of the trial, which translated out in a just hazard ratio of clinical worsening of 2.0. The study had limitations, including a small sample size, selection bias, in that this population is from a randomized clinical trial population with its own inclusion-exclusion criteria. We expect there is some residual confounding, and the relative protein abundance by nature of the O-link processing does make some of the analyses more complex. But in conclusion, we found that higher plasma endostatin was associated with shorter 6-minute walk distance, lower physical activity, and more clinical worsening. And really what we're looking to do next is to model the longitudinal trajectory, see if we can assess response to therapy, and via those understand its potential as a biomarker for disease progression and potentially in remote monitoring. So with that, I want to say thank you to all of my collaborators and mentorship at Penn, collaborators through Phantom, as well as in the UK for our validation cohort. And that is all I have. Thank you.
Thank you, Amber. Excellent presentation. Thank you very much. Dr. Zamanian?
Hi, Amber. Great presentation. Really exciting work that you're doing. So I have a sort of a 2-part kind of question. First, I would love to hear about, have you thought about the analytic approach to your longitudinal models and how you would think about that from a modeling perspective? And then the second, the more important one, I think really is, we all have biases in selecting specific biomarkers that pique our interest. What you're showing here is not only one biomarker, endostatin potentially has the highest relationship to walk distance in your cohort, but what would a panel of biomarkers and analyzing that a panel rather than a single biomarker do? Do you think that would be a more powerful or a less powerful approach? We know that there is tremendous heterogeneity in PAH, and it is going to be unlikely that one single biomarker is going to have that profound relationship to exercise tolerance. So I'd love to hear your thoughts about those.
Thank you. To your first question, I have thought a lot about how to longitudinally model it, and I think we're going, just by nature of power, we're looking more at like mixed effects models, although I wish we had more data so we could do like maybe a more sophisticated clustering model or trajectory analysis. So if you have any insight into how to do those, my numbers, I would be intrigued. And your second question, I know that endostatin has been additive in panels, biomarker panels, and so I expect that may be how this is useful. Other things, I mean, I was also interested in several of these other proteins that I found to be significant. For instance, these HSPG2 and insulin growth factor BP7. I know there's data for both of these elsewhere as well. And so I would be interested in how they function as a panel. I'm also interested in how they differentiate patients and what each one of them represents, and maybe they could help us basically sort patients into different phenotypes, which is also, I know, a hot field.
So thanks very much. I'm going to apologize before I ask my question, because you've probably answered this question when we've spoken before, but it's going back on Roham's point about the longitudinality. And I just wonder if you can just remind us how you thresholded your actigraphy data. So obviously with the 6-minute walk, you've got a single time point, so it's straightforward. But could you just explain to us how you dealt with that kind of actigraphy data in terms of how many days did you average, or how did you do it?
So just this, I guess this is all baseline data, but it is longitudinal in that the baseline physical actigraphy data is 7 days long. And so it is a continuous value of added, like just additive vector magnitude over those 7 days. So it's a pretty like simple approach. Some of the people in our group have done more sophisticated PCAs, and I'm sure there are more creative ways to deal with it, but that's what we did just to maintain the continuous nature.
Okay, great, thanks. And then, yeah, I can't remember again from when we spoke before whether you have longitudinal protein samples, but I mean, I can share some methods, but we did do some mixed effects trajectory models on actigraphy data and COVID stuff before. So I'm happy to kind of share that if it's useful going forward, but it needs probably at least 3, if not 4 time points of samples, and I can't remember what you've got.
Yeah, we do have 4. So actually that would be amazing. I appreciate it.
Okay, great.
And Josh Vessel has raised the question about validating your findings, and he asked whether you have had the opportunity to validate or test your findings in the PVDomics cohort and data set.
We have not. We've considered it as a source, but we have not applied. I would like to.
Well, thank you so much, Amber, for your presentation and for the questions. So we need to move on to our next presenter. We are next having a presentation from Abdelrahim Sara from University of Lorraine in France. And Abdelrahim, go ahead.
Okay, so hello everyone. My name is Sara Abdelrahim. I'm a PhD student from the University of Lorraine, France. And today I'm going to present for you my abstract entitled Von Willebrand Factor-Mediated Alpha-V Beta-3 Integrin Signaling Drives Pulmonary Artery Smooth Muscle Cells Proliferation in Pulmonary Hypertension. Briefly, as you all know, PAH is considered as the first category of pulmonary hypertension. It's a rare and progressive disease that affects small pulmonary arteries ranging in size from 50 to 500 micrometers in diameter, and the increase in pressure in these arteries leads to increase in the pulmonary vascular resistance to a value higher than 2 Wood units. Now, having a closer look at the vascular level, the 3 main layers of the vascular wall, intima, media, and adventitia, are all structurally altered in PAH, where we have thicker wall, narrower and obstructed lumen, and we also have higher cellular proliferation, mainly of the smooth muscle cells in the media and the endothelial cells in the adventitia. We also have infiltration of inflammatory cells and fibroblasts contributing to inflammation and fibrosis. Now, one important mechanism involved in this pathogenesis, and it's a key event in PAH, is endothelial dysfunction, and it disrupts this normal balance between vasodilators and vasoconstrictors. It also involves this endothelial to mesenchymal transition. One important marker of endothelial dysfunction is VWF, or von Willebrand factor. It's a blood glycoprotein primarily known for its role in homeostasis, but in the context of PAH, VWF is released from its storage sites in the Weber palette bodies of the endothelial cells, and it moves either towards the bloodstream or towards the underlying smooth muscle cell layer. In the plasma or in the blood, the high levels of VWF has been associated with higher risks of death and lung transplantation. However, at the level of the vascular level, we do know that VWF is highly expressed in the media and in the intima region of the vessel, but we don't really know its role in vascular remodeling. This question is very important because VWF appears to have a very important role in other vascular diseases, notably in atherosclerosis. And for example, as you can see here in the photo, we have a murine carotid artery ligation model of atherosclerosis, and we see how the media is very thin in the VWF knockout mice compared to the wild-type mice. Not only that, but also VWF at the molecular level appears to induce vascular smooth muscle cells in the systematic circulation via an integrin, which is a cell surface protein named alpha-V beta-3. Interestingly, this alpha-V beta-3 is highly expressed in the pulmonary arteries of PAH patients, and also it's known to activate signaling pathways involved in smooth muscle cell proliferation, exactly like this pulmonary artery smooth muscle cells proliferation. So my hypothesis was that VWF could promote pulmonary artery smooth muscle cells proliferation and phenotypic modulation, and I also suggested that probably alpha-V beta-3 may mediate this VWF-induced cell response. Due to the time limitation, I'll pass directly to my results. So first of all, I assessed cellular proliferation, and my results monitored higher cellular proliferation following VWF administration compared to my positive control PDGF and to my negative control. Also, cellular viability was high and was increasing over time compared to my PDGF and control-treated conditions. Then I went to check whether the alpha-V beta-3 has a role in this, and to do that, I inhibited the alpha-V using siRNA, and what I have seen is that the increase in cellular proliferation observed in my control condition, the scramble siRNA1, following VWF treatment was decreased as I inhibited the alpha-V siRNA, alpha-V, sorry. And this was also confirmed by another type of inhibition, which was the peptide inhibition, so indicating that the alpha-V beta-3 is implicated in the VWF-induced cellular proliferation. Then I went further to check whether this proliferation was also due to a change in phenotype. So for that, I selected 2 contractile markers and 2 synthetic markers that are known in PAH, and I went to check for their protein expression of these markers. And what I have realized is that VWF is decreasing the expression of the contractile markers, and it's increasing the expression of the synthetic markers, indicating that it's promoting a contractile to synthetic phenotype switch. Now, does alpha-V beta-3 has a role in that? To do that, I did the same protein analysis, but this time using inhibitors of alpha-V siRNA, and what I have seen is that the decrease in the VWF-induced contractile phenotype was restored once alpha-V was inhibited. In the contrast, the synthetic phenotype was decreased again once alpha-V was inhibited, indicating that alpha-V beta-3 is implicated in the VWF-induced phenotype switching. Following that, I went to assess the signaling proteins that could be activated in this pathway. So I selected the main signaling proteins involved in cellular proliferation and modulation as FAK, SRC, AKT, and MAP kinase. And I exposed my cells to VWF for a 1-hour interval, and then I tested the phosphorylation in control conditions and alpha-V inhibitor-treated conditions. Just to mention that in the first photo, as you can see, the phosphorylation was attenuated in the conditions treated with the inhibitor compared to my control condition, and this was true for all the signaling proteins studied. So as a conclusion, I was able to find that VWF could interact with alpha-V beta-3, activate downstream signaling pathway that are probably involved in transcription factor, and as a result, lead to smooth muscle cells proliferation and phenotypic modulation. Thank you so much for your attention, and I'm ready for questions.
Thank you very much. And we'll open the discussion with Elena. I see a raised hand. Please go ahead.
First of all, it's wonderful presentation and very thought-provoking, to be honest, because Wall-Wille-Brown factor been here for ages, right? And the mechanisms are not very known. So I have, in fact, 2 questions. One is philosophical and another one is philosophical too. So first question is, so it's very highly likely that von Willebrand factor plays a role in disease initiation, right? So because it induces synthetic phenotype. But just what happened, like which role it plays, if any, in disease progression when smooth muscle cells already phenotypically remodeled? And my second philosophical question is, do you think von Willebrand factor will affect other cell types, like, for example, fibroblasts, macrophages, signal back to endothelial cells, like promoting endo-MT?
Yeah, so concerning, I will start with the second question because it's easier a bit. So whether it affects other cell types or not, actually we have to check. So I don't have the answer. We have to check, especially that, yeah, integrins are not only expressed by smooth muscle cells, so they are expressed by other cell types, especially endothelial cells. So maybe it contributes, but we have to check. I don't really have the exact answer to that. And concerning the first question, whether it's important or not, because already the vascular remodeling is there, I think it's important because we don't really in PH, as you know, we don't really know how things proceed, like how things start. So whether endothelial dysfunction is at the beginning or whether it's part of the different mechanisms taking place, so we don't really know. So I think we have also to inhibit in order to see. So we have to try to go to translational steps later on in order to inhibit and see whether it affects positively or not.
So I will be looking forward to your animal reversal experiments. I hope you will present it at the next ATS meeting.
Yeah, I hope to. They are ongoing, and we are using now the inhibitors. So I hope I'll get nice results to present for you next year.
Thank you, Abdel Rahim. Thank you very much. We also have a question, Dr. Lawrie.
Yeah, no, thanks. I agree with Elena. It's very thought-provoking. I guess a couple of things come to mind. One is VWF is a very sticky, long, big glycoprotein that can bind to lots of other things. So I guess in thinking about those disease initiation, disease progression stages, I wonder how much is primarily driven by VWF. And I guess then technically from your own experiments in cell biology, for example, I just wonder how you controlled for the different dimers and multimers that can occur with VWF, and whether there's a difference in your models, whether you have small dimers versus the big long multi-chains kind of multimers. I don't know if that's something you've looked into.
Yeah, actually, this is a perspective of my work, because till the moment we know that there's an interaction VWF alpha V beta 3, but exactly how it's going, which dimer is involved, which site is involved, we don't know. So this is always also in the perspectives of the work. So yeah, we are working on that in order to find which exactly dimer is responsible for the binding with our protein of interest and involved in this effect, of course.
Okay, so I guess everyone likes to talk about their own work. So I guess one of the reasons for asking is that we work on a protein that we know binds to VWF. It's stored in Valdopa bodies with VWF, and it drives a lot of the same signaling cascades that you've described there. So I'm happy to follow up with an email afterwards, but it'd be interesting to know whether if you blocked that antibody, whether you saw the same effect and so on.
Yeah. Actually, at the systemic level, the A2 domain of VWF appears to have a positive effect on cellular proliferation. So probably if it interests you, you can check more this domain specifically on VWF. But our experiments are ongoing in order to find exactly what domain is involved in pulmonary artery smooth muscle cells.
Great. Thank you very much.
Welcome.
Excellent. Thank you, everyone. So I think for the sake of time, we also need to move on to the next presenter. Our next presenter is Megha Talati from Vanderbilt University.
Thank you for this opportunity and for selecting this for this small webinar. So I am at VUMC, Vanderbilt Medical Center, and Research Associate Professor. And this is one of the projects that I have been working on. And I'm just going to introduce this small little RV-related dysfunction, RV dysfunction in PH, shifting from lung to the RV. So we just recently published that lactic acid is increased in PH patient, so in circulation of PH patients. And as you can see, when we looked at the relative units, these are relative units, there was a trend towards increased lactic acid in circulation in PAH. And when we looked in the animal model where we had done some metabolomic studies, we were able to show that the end product of glycolysis, which is pyruvate, was increased, and there was also increase in the lactate, where pyruvate gets converted to lactate. And we saw an increase, though it was a modest increase, there was an increase in this animal model of PAH with BMPR2 mutation. So we just looked at this lactate dehydrogenase enzyme and in the H9C2 cells, and these are the cells which are cultured cardiomyocytes, and we were able to show that when you introduce a BMPR2 mutation in these cells, there is increase in this lactate dehydrogenase enzyme activity. And when we went back and looked into the PAH samples, RV samples, we were able to show that this activity was also increased in the human RV. The other point I want to make before I move forward with this, so we were able to show there is increased lactate in these mouse model RVs. But interestingly, pyruvate can enter the TCA cycle, but in our model, the mutant model, we were able to show there was increased citrate, but the downstream TCA intermediates were decreased relatively. For example, succinate, although not the other two statistically different, but succinate was decreased. So what does this lactate do? It does not, I mean, there is increased lactate. So there was something that I wanted to pursue. So first thing we looked at was what can lactate do if there is increased lactate in any cells. So basically it can cause epigenetic modifications by serving as a donor for lactylation of lysine residues. Under normal physiological conditions, this lactylation can help maintain the structure of a cardiomyocyte. Also functionally it is important. But if there is an imbalance between a glycolysis and a TCA cycle, it can cause reprogramming by increasing the histone lactylation. And there is also non-histone, which I'm not going to talk about right now, but it can cause histone lactylation, which was the first thing we wanted to look. And in context to our PAH, it was shown that lactate can cause histone lactylation and it can promote cardiac hypertrophy, as well as ventricular remodeling in the stack model of mouse, as well as in pulmonary endothelial cells. They can cause, pulmonary smooth muscle cells, they can cause proliferation and vascular remodeling in an animal model. So we just came up with this hypothesis that if there is increased intracellular lactate, which is a product of aerobic glycolysis, it can promote lactylation in the failing RV cardiomyocytes. So what we did was, just going briefly over the methods before I dive into my results, we just use immunoblotting, very basic techniques, to see whether there is increased lactylation in the human tissue, in a mutant mouse model, and in cardiomyocytes. And that immunofluorescence, we wanted to see where exactly it is, and then just do mass spectrometry to show whether there is increased lactate. And the last thing we did was a lactate assay in the cell culture. So the first thing I want to tell you is that there was increased lactate protection by mass spectrometry. First, we wanted to see whether this increased lactate that we see in circulation, where does it come from? So our data from this animal model of BMPR2 mutation showed that the increased lactate, which is there in plasma, is predominantly coming only from lung and RV and not from any other tissue, which was assuring, because in PAH, these are the two organs that are important. So the second thing we looked at was what happens to this lactate, which is there in circulation, because lactate is also there in the RV cardiomyocyte. There are two monocarboxylic transporter molecules, MCT1 and MCT4. MCT1 can let the lactate go in and out, whereas the MCT4 only secretes the lactate out from cardiomyocyte. So we looked for the expression of these two transporter molecules. And as you can see in the pH samples from human tissue, we were able to show there is increased MCT1 both in IPH and HPH patients, and this is from the RV cardiomyocytes. And MCT4 was increased only in the IPH and trended higher in the HPH. And when you look at MCT1 and MCT4 by Western, this was the localization and this was Western, we were able to show that MCT4 was increased in the PAH patients, but not MCT1. This was the whole tissue. So next we looked at was what, So next we wanted to see whether if there is increased lactate in these RV cardiomyocytes, does it increase lactylation? and we were able to show that there is increased lactylation in the PAH sample from RV samples compared to controls. And when we looked at the mouse model that we have for BMPR2 mutation, there was a trend towards increase in lactylation in these RVs. And when we looked for the localization localization of lactylation residues by using this lactyl lysine antibody, as you can see, there was increased lactylation both in the HPH and the IPH patient RV tissue. So the next thing we wanted to see was what if there is free fatty acids in the circulation, whether it would influence lactate production, because we have shown in the past that there is increased fatty acids in the patient circulation. So we used 2 different concentrations of palmitate, and these studies we did in the H9C2 cells which have BMPR2 mutation, and we have 2 different mutation cell lines, and as you can see, the endogenous lactate was increased early on, whereas the secreted lactate was increased later on in these cells. And there was not much of a difference with different concentrations of palmitate used, suggesting that there is lactate produced by RV cardiomyocytes or cultured cardiomyocytes with a BMPR2 mutation. So, and when we looked at whether there was increased lactylation in this cell, in these mutant cells, we were able to show that the lactylation was increased after 48 hours within these in these cells with BMPR2 mutation. So to conclude, we were able to show that the elevated plasma lactate likely originates from these 2 organs, which are pH related organ, lung and RV, and can cause both local and systemic adverse effects. And in human PAH-RV, altered cardiomyocyte metabolism due to increased lactate can promote epigenetic modification through lactylation of lysine residues. And this is just a schematic showing that MCT1 may be able to transport lactate in and out of the cell cardiomyocyte, whereas MCT4 may be just secreting it out from these cells. And these lactate molecules that are increased lactate that is present, metabolite that is present in the cell, can cause both lactylation of histones or may also have a potential role in non-histone lactylation. And I would like to thank my mentors and lab members, as well as the funding and the patients and the donors. Thank you.
Thank you, Dr. Talati. Nice to see you again too. Dr. Zamanian.
Hi, Megha. Very nice presentation. Really, really interesting data. Thank you for sharing with us. I have an overarching question. We know lactic acidosis is not specific to pulmonary vascular disease. Can you talk about the implication of your findings? Is this part of a deconditioned state? Does this have relevance to left heart failure? Tell me about the specificity of this as it relates to pulmonary hypertension.
I'm trying to go back. I'm not able to see anybody right now, unfortunately.
We're seeing your beautiful desktop.
I think you should stop sharing.
Yes. Oh, okay, because I have to stop sharing. Yeah.
But sharing is caring. But
yeah, sharing is caring. Okay. So acidosis, the thing, I have to say that we had one of these. Anna Hemnes had one of these metformin trials where they had shown there was not much of lactic acidosis there, lactic acid acidosis, because I was interested in knowing whether metformin reduces that, and there was not much of an effect, is what I would say. So I'm not sure how increased lactate is going to have any relation with acid lactidosis, because at least in the trials that we have done, we have not seen any changes in that in the PAH trial with metformin. So there was not increase or decrease with lactic acid acidosis. So what I'm trying to say here is what I think lactate is doing is it is trying to modify, bring about epigenetic modifications in a cell. It can be a RV cell or it can be any inflammatory cell. And I'm just diving into something different right now, just because it has been shown that lactylation occurs in inflammatory cells and mainly in macrophages. And so I'm trying to say here that somehow lactate is playing a role in modifying these cells more than causing lactic acid acidosis, if I'm answering the question correctly. So more than circulation, it is doing something to the cells, to the organs individually or the cells individually, not systemically. Is my take on it right now, because this is a very preliminary stages project, which is we have just started working on this a year ago, and because we saw these new findings, we just wanted to try and understand what is lactate doing.
So the implication of your work, sorry, I'm a
Oh,
clinician.
sorry.
The
Yeah.
implication of your work is not so much that there is elevated lactic acid in patients with PAH, but that lactic acidosis has a downstream impact.
Correct.
And so that continues my question is that we see lactic acid elevation in sepsis, we see it in heart failure patients, we see it in poor end organ perfusion. What would be really interesting for me is if you had a comparison and contrast of right ventricular responses or myocardial responses to that lactic acid in other disease types. How is this pathway relevant? Because ultimately, as a clinician, I would want to do something therapeutically to help the patients, right? That's the next step of your work. And so I would want to know, how is this very specific to pulmonary vascular disease, PAH, right heart failure? Or does it have a global role to treating sepsis, heart failure patients, and, in the ICU, etc.?
Yeah. Yes, and right now I may not have an answer to that, because I'm looking at it from a one disease perspective and not a global perspective, because this was a finding in our disease model where we are trying to explore and understand what it is doing there. But you may be right, I think, that if it is going to be present in other diseases too, it may have implications there as well. I don't know which cell type it may affect there, what is important in that particular disease, because if I'm studying PAH, I'm looking at the disease and the organs that I'm interested in. If you are studying a different disease, it might be in context to how it is affecting that disease progression, and how would you intercept it with a drug to reduce that it will improve the condition. So this is regulated by histone lactylation. Correct. So this is what I'm trying to do. I have not looked at the targets downstream of histone lactylation yet, because I'm not yet sure. I just want to first find out whether it is a histone lactylation or whether it is a non-histone lactylation. So we are right now having a hint that it is showing it may be histone lactylation, and to be more specific, and that's why I did not present the data here, even though we have some hints, it could be 2B or something to do with one of the 2A, 2B histones, but I'm not 100% sure. So if once we get convinced whether it is that it is a histone lactylation, we move down to the targets of that specific lactylation histones, and not go first to the targets before knowing what is being lactylated in the RV. And we are looking at inflammatory cells as well, because that would be a different set of genes that would be affected, and different set of lactylation products too. If I'm answering your question.
Thank you again. We appreciate it. Thank you so much, Dr. Talati. So we need to move on to our last presenter, Adriano Tonelli from Cleveland Clinic.
Perfect. So I'm going to be quickly talking on a transcriptomic analysis we have done in endothelial cells and smooth muscle cells of patients with pulmonary arterial hypertension to understand treatment pathways that will help us in the future personalize treatments for pulmonary arterial hypertension. Pulmonary hypertension treatments are mostly vasodilators with a new agent now that targets the activin BMP pathway and acts on some disease-specific abnormalities. The treatment of pulmonary hypertension is based on disease severity and not on mechanisms of disease, and understanding that would be important for treatment selection and also for comprehending the potential crosstalk between pH-specific pathways. This is a slide just to show that the new treatment, sotatercept, that acts on the activin BMP pathway interacts with other known pathways in pH, vasodilatory pathways such as nitric oxide, endothelin, and prostacyclin in different ways. I'm not going to go in details for a matter of time. But the problem with understanding this crosstalk and the different treatment pathways on the cells is that they're hard to get. And most of the cells we're getting at the moment are coming from explanted lungs at the time of lung transplantation or at the time of autopsies. We have recently developed a protocol to isolate circulating endothelial cells from blood obtained from the pulmonary artery. And we have more than 100 now colonies where we can test different pathways related to pH treatment. This is how we do it. We during the RAHA catheterization, we collect blood, we isolate the Buffy coat, then we lyse it, we wash it, and then we culture in endothelial cell media. We've previously shown in this paper that the cells do express CD31 and CD144. They form tubes similar to what cells that you get from explanted lungs, pulmonary endothelial cells, or cells that you buy commercially. For the purpose of this study, we performed transcriptomics in circulating endothelial cells, in pulmonary artery endothelial cells, either from pH patients and controls, as well as smooth muscle cells. And why it's important to look at these? Because it can help you understand cell-specific mechanisms, dysregulated pathways, early molecular changes that haven't really translated yet in structural remodeling, crosstalk between cells, and hopefully enable precision treatments. This is the transcriptomic results of our circulating endothelial cells, the pulmonary artery endothelial cells, and smooth muscle cells. This is comparing pH with controls in each of these plots. And as you can see on the box below, in the table below, 566 transcriptomes were more than twofold different, significant more than twofold difference between pH and controls. The same with pulmonary artery endothelial cells, 919 different from pH versus control, and smooth muscle cells, 1,000 or more than 1,000 between pH and controls. We put all this information together and we identified 33 coding genes that were significantly different and the same in all of these three different comparisons. So 33 of these transcriptomes overlap when comparing pH versus control in these different types of cells. And these are the genes, the expressions, and the one table on the left is showing the ones that were commonly affected in the same direction, more than 2 folds in all different cell lines when compare pH with controls. And on the right, you could see that the endothelials and the smooth muscle cells do go in opposite direction, but they are significantly different. We noted that, for example, HAN2 and NKX25 that are involved in vascular development are reduced. TBX3, regulator of tissue development, was increased. Noggin, FGF5, UPA were increased. The same with platelet-derived growth factor. And now a very exciting molecule that's associated with the new treatment pathway, FSTL3, follistatin-like 3, was also elevated in endothelial cells. Part of the transcriptomics, we can now look at different parts of the treatment pathway. And as you can see in the nitric oxide prostaglandin, BMP activin, and nitric oxide prostaglandin, there's a bunch of transcriptomes that are abnormal, and they can certainly be indicators of the function of these important pathways. So this is a cartoon showing you our most significant findings, what we saw in the circulating endothelial cells, as well as the pulmonary artery endothelial cells. The patients with PH have decrease in the caveolin 1, increase in phosphodiesterase 5 that will obviously destroy cyclic GMP. The caveolin 1 decrease will reduce NO production. In the prostaglandin pathway, we saw a decrease in the receptor for prostaglandin receptor, as well as an increase in other receptors that are associated with EP2, which could represent a mechanism of how side effects develop in patients that receive prostacyclin, headaches, diarrhea, nausea, vomiting. And in the BMP activin pathway, we saw a decrease in BMPR2, increase in activin receptors, increase in the activin, increase in noggin that binds BMP, all of that potentially leading to cell proliferation. So in conclusions, our findings shows that we noted expression differences between the cells from controls versus PH patients, both in circulating endothelial cells, pulmonary artery endothelial cells, and smooth muscles, that suggesting that the circulating endothelial cells, which are easily obtainable from patients at the time of RAHA catheterization, may be a good model to investigate treatment pathways or other pathways of the disease. And these investigations may aid in treatment, and as well as better understanding the pathology of pulmonary hypertension. And we notice a complex interplay between these pathways, that there might be a significant crosstalk between them that might also be important for treatment selection. In this gene expression alteration will give you opportunities for identifying biomarkers and selecting treatments. And that's it. Thank you.
Thank you, Adriana. Thank you. Dr. Lawrie?
Hi, thanks very much, Adriano. I guess 2 quick questions. One is, I guess, the obvious one, is just where do you think these cells are coming from? And the second one is more of a methodological thing. And I wonder whether you've thought about trying to apply some of the kind of deconvolution tools from single cell and various other things to do a bit more of a deep dive comparison into the different endothelial cells to see whether they're more like a particular subset of endothelial cells than the other.
Yeah, that's an excellent question. Is kind of the holy grail, but I look at it in a different direction. It's like, yes, the cells, obviously I get blood from the pulmonary artery, but are the cells really only coming from the endothelium of the pulmonary artery, or are coming from any other circulation, parts of the circulation. But they do express similar signals and abnormalities as the pH cells that we isolate from the lungs at the time of lung transplantation. And so, yes, they may not be exactly coming from the endothelium, may not be exactly the same as the pulmonary artery ones, but they do carry the pH signal, and they may certainly be a useful model, even when it's not exactly coming from the pulmonary artery in the worst case scenario. So, and yes, single cell will be a good next step to work on.
Dr. Zamanian.
Adriana, great presentation. So let me ask you to be a futurist, and tell me, how do you apply this as a companion diagnostic to select therapy in patients? So walk us through that. There's going to be a lot of heterogeneity of the cell types, etc. But imagine you're 10 years, 20 years down the line. We all have some chip implanted in our brains, and there's AI, whatever. But tell me, but functionally, how do you standardize something like this, and how do you apply it to patients?
Yeah, that's also excellent question. The good thing is that we have a recovery rate of approximately 80 to 90%. We have been optimizing the model. So imagine a patient that you see in the clinic has cells growing in the lab at the same time, and you can certainly be testing things. Yes, at the beginning, it's obviously a broad approach of transcriptomics, very generalized approach, until we identify key molecules, key factors that then we can individually test in a very specific test. For example, prostaglandin receptor it's a key thing, PGI. Great. Or we need to look PDE5, or we need to look at some other specific molecule, activin, or we look at FSLT3. And that will now create, as you said before, a panel of key molecules in each pathway that will then guide you to treatment. Exactly. We cannot really do transcriptomic for every patient because obviously patients need to be treated right away. We cannot really be waiting. But also, let's say you wait, you do these, you could also potentially tell the patient you're not responding to treatment as expected in a patient that, let's say, has suboptimal response because you see this is your panel. Different pathways are affected different way, and we have no good medication for you just yet, or you need to have a combination of these 2-3 medications. So I think that could be a potential patient that doesn't really improve on therapy, or by the time hopefully we identify key molecules that we can specifically check rapidly and not doing this culture the cells and then do transcriptomics.
There's one question from the Q&A. So, like, supposing that you could continue on one of the targets or like a number of targets, do you have any preferences? Or I think there are a number of established targets, but how would you move forward in terms of either diagnosis, so serving as a biomarker or as a treatment target?
We are opening to all kind of discovery. Obviously, when we do these, we will have all the hemodynamics on the patients, all the risk scores, so the survival status, the outcome measurements, where we can identify, oh, look at this great pathway. It could be a different pathway, not treatment related just yet. So we're open for that discovery. We haven't really identified a good biomarker at this point. I mean, I don't want to say the maybe few things that, like, for example, erythropoietin. There are things that are very interesting that we have seen in some of the data that we need to explore further for biomarkers. But mostly focusing on treatment. And this is one of the approaches we have for treatment selection. We also use iontophoresis of medications in the skin, but this one, I think it needs more investigation, and we are ready to send like 28 patient samples with different transcriptomics that will give us a good idea of what's next.
Perfect. So there are no questions left, no raised hands left. So I think it is time either to continue work or a football time for others. So I would like to thank all the speakers for their input today, and as well as the panelists for their great contribution to the discussion. And Mariam, thank you very much for organizing this in a fantastic way. So have a very good day altogether.